damage the food. So that is the basic summary. So now let's talk about food. I have talked about that the 1/3 of all manufactured food is world is spoilage or is lost. So why they are lost? Because of the microbial content. What do we mean by the microbial content? Microbial content means some microorganisms like bacteria. Most of the food damage is done by bacteria and fungus, and some of those molds, we can say, in those fungus generate those molds which will damage this really qualitatively and quantitatively in both the directions. Okay, so if we are talking about the food damage or food spoilage, we want to talk about something about the shelf life. So before going to that, why this microbe will damage the food? Because they also need food like us. They also need protein, lipid, carbohydrate like us. So this is PLC, this protein, lipid, carbohydrate, these parts or these are the macromolecules that builds us, and these are the macromolecules that build everything, like every living organism like bacteria too. So they also need protein, lipid and carbohydrate. So we have our food, we store that food for our helping purposes to taking this protein, lipid, carbohydrate from them, but bacteria took toll on it and they snatch our food, they eat our food, and they make up. And when they eat our food, they stay in our food. They stay with the difference between them. They are really small, and the food particles we are storing is really huge for them. So they are actually they are present in those food particles to damage them. And when they are damaging them, so whenever a food particle, for example, in this background picture we have seen in this apple. Whenever this food particle, this apple is affected by different bacteria, this apple is damaged, and those bacteria will present in the apple. And whenever it happens, we need to discard that food because we don't have any other choice. We do not take a chance to eat this food further because we know if we eat this food, it can make us any damage. Why? The question is why? Why it's a damage for us if we eat this bacterial eaten food? Because bacteria are still present in many vigorous amounts. For example, billions of bacteria present in this food to make this food characteristic appearance like that. If we eat this food, that can bring us diseases. How can they bring us diseases and why? Because those bacteria can cause diseases, as we know. How bacteria cause diseases? Bacteria cause diseases by many ways. They can secrete some of the proteins which are harmful for us, which we call toxins. So when we eat those toxins go into our body, and sometimes the toxin acts on our intestinal tract, and sometimes they can attack our nervous system, sometimes they can attack our muscular system and all these different systems. So we want to prevent this damage. That's why we need to discard those foods, and that's how the food is lost. So now let's talk about the shelf life of food. So what is a shelf life of food? So some of the food are normally what those foods are not favorable for bacteria. So suppose there are different variety of foods. Some of those foods are really favorable for bacteria to attack on to lean on, especially those foods which are full of protein contents like eggs. Those are called perishable food for bacteria. They just come in and have fun with in those cases. Okay, so egg and for example, let me tell, fish, meat and all these things will be that. Though egg has a covering, but still it is a very perishable food for bacteria because it is delicious. It has protein, it has lipid, it do not have carbohydrate, but it has protein and lipid, which is really important for the cellular growth and division. And second thing is a semi-perishable food. What do you mean by semi-perishable? Well, the name suggests is all that these semi-perishable foods are not that much favorable like perishable, but it is favorable for bacteria. Like bread. Bread is actually hampered by not only bacteria, it is hampered by molds. There are bread molds out there, which you find in bread. When you leave bread for some time, you can find this greeny skin velvet color appearance come into this bread. And this is why to different mold attack into the bread. Okay, and if we look at and the final one is a non-perishable food. Normally those foods which are not favorable for bacteria do not attack those foods normally or generally. Those are called non-perishable. For example, pasta. It is so dry, the water content is so low, and it is not attacked by bacteria, so that is non-perishable. Why our food's perishability depends on different things. So what is the shelf time? Shelf time is the time for a food to be safe and to be as it is in its primary characteristics. Okay. So the perishable food have really short shelf life. Semi-perishable have a little bit bigger than the perishable food, and the non-perishable food have the longest shelf life. Okay. So if we store if we store meat and also pasta individually, not in refrigerator, in outside the refrigerator, in normal room temperature, the first thing to be damaged will be that meat, not the pasta, because meat is a much more perishable food. Okay, so this is the basic concept about perishability. So what are the basic conditions that is triggering the food spoilage? Well, the conditions are water content of the food, the pH of the food, the physical structure of the food, oxygen, and the temperature, because all these things are correlated with the growth and division of bacterial cells. Bacterial cells needs water to grow, because every single living organisms need water to grow. So bacteria needs too. So bacteria needs water. That's why the water content. If a food have a lots of water content, they have a high probability of catching those bacteria into this food. So though there will be really perishable foods. So fish, meat, and all these things have lots of water content in them, because you can find it when you cook a meat or fish. You cook a meat, then you can find the water is coming out from it. Okay, because it's a tissue. Tissue contains water, as we know. Now let's talk about pH. If the pH is lower, we have a optimum pH. Not everyone or every organism have an optimum pH to work with different enzymatic reactions which is going on them. For example, bacteria have an optimum pH. So this optimum pH for bacteria are normally 6 to 7. So this is the normal pH or near neutral pH. In this near neutral pH, all the enzymes in the bacterial cell works properly. So they want food which have the pH of 6 to 7. Then they can grow and divide really rapidly. So normally, and in case of molds, this pH variations is little bit of acidic. So they can grow in acid contents like 2 to 8. So their actually window is really higher for molds. They can grow in acidic content and slightly in basic contents too, but they are favorable the growth in the acid contents. Okay. Normally below 4 or 3 pH we cannot find bacteria to grow because bacteria for a hard time to grow in the pH of 4 and 3 or a little bit below that. That's why the acidic food is not a good choice for bacteria. Acidic food is a good choice for molds because they can grow in acidic content like pH 2 and 3. That's why it's a good choice for them. For example, we have the acidic foods like this. In this case, this tomato and this lemon, and these are the acidic content because we have acids in them. So these food are not generally attacked by bacteria; they are attacked by different molds. You can find in lemon. If we cut the lemon and leave it like that, you can find it. The molds will grow up like a velvety structure of molds. You can find it. Okay. Okay. Now let's talk about the physical structure. If you want to talk about the physical structure is also important. But if you have a hard structure, if you have a really strong structure, if you have a really hard sheen on in the background of it, that will be really difficult. That will be really difficult to enter. For example, these dry foods here, the flour. So these are not generally been attacked by bacteria. So we can store them for a long time. They do not have the water content and they are really hard. So if you have a watery part, that will be really helpful for bacteria, because bacteria can swim from one place to another place. They can enter really from one place to another place. That's important thing. And most of the foods contain water, as we know, because if we don't drink water, but still we can find water from the foods because foods are having water. There are not some of these extremely dry foods out there, but we generally do not use water, and we use to make this extremely dry food watery when we eat it. Okay, so that's why physical structure is important. And oxygen, and the presence of this oxygen is also important, because oxygen is needed to carry on the cellular respiration, as we know. So oxygen is needed. That's why if we have food, those are stored in anaerobic condition. Actually, food have oxygen because food is exposed to all the time in the oxygen. So if we store them in anaerobic condition, like in a can, or in a can like this, which is a yogurt is packed, this anaerobic environment, we do not find some of those bacteria can enter into this. Why? Because oxygen is needed for many bacteria to grow, but we also have bacteria that do not need oxygen to grow. They are anaerobic bacteria, and those anaerobic bacteria can grow in without the presence of oxygen. Okay. And finally, the temperature. Temperature plays a key role, is a big role in the division of bacterial cell, because optimum temperature, as we know, in case of bacteria and molds, there are optimal range of temperature to grow on. For this in this temperature, the bacterial cell will grow and divide rapidly. So the optimum temperature normally is the room temperature, and for the many organisms it is 37 degrees Celsius temperature, where we used to incubate them for our study purposes in laboratories using incubator. So normally the range will be for 25 degrees Celsius to 40 degrees Celsius. This is a good range for bacteria to grow, to have a growth. So normally in India we can find this growth temperature in summers, because summer in India the temperature rise to 45 degrees Celsius temperature, and that's why in summer the foods are really spoiled really quickly, because in this growth range we have different types of bacteria work together. Most of the bacteria can be active in this range, and we call those bacteria as Mesophilic bacteria. So what do you mean by Mesophilic bacteria? Mesophilic bacteria love to grow in between this window of temperature 25 degrees Celsius to 40 degrees Celsius. But if you store them below 25 degrees Celsius temperature for different countries, if you store them, but still they can be spoiled, but not by these Mesophilic bacteria. There are different sets of bacteria they can spoil this below 25 degrees Celsius temperature. For example, 5 degrees Celsius to 15 degrees Celsius. In this temperature range, there are different bacteria to grow, and those bacteria are called Cyclophilic Bacterias, or sometimes they are called Cyclotrophic bacteria. The Cyclophilic Bacterias can grow in relatively lower temperature than those other bacteria, and those bacteria can also spoil foods. So actually they are not destined to spoil food, they actually come in to eat food. So they eat food, but this our problem, that we can't eat those foods which are what you can say contaminated by bacteria. If we find bacteria in our food, we need to discard that. That's our choice, and I recommend you to do that as a microbiologist. Okay. So these are the conditions that controls the growth of microorganisms in the food, or you can say these are the conditions of food spoilage. So how can we, if we manipulate these conditions, really tightly, then we can stop spoilage. For example, if we release the water, we can make the food dry. That will be really lower chance for spoilage. And after drying the food, if we make the food in a little bit of acidic pH, for example vinegar, for example different sauces, they are not normally caught up by these bacteria, because the pH content was really lower. 1 to 2 pH is really hard time for bacteria to go and enter and divide in this pH. And physical structure is not plays that much key role, but oxygen is also important. So if we make those foods canned in an anaerobic condition, we can prevent the damage of those foods by those bacteria, by those aerobic bacteria. But the anaerobic bacteria are there. And also the temperature. So the main key event to manipulate it during this food preservation is the water content, the pH of the food, and also the temperature of the food. If we control these regions, this part of the food to do for temperature control, we have refrigerator. If we store this refrigerator, normally in refrigerator the temperature is 5 degrees Celsius or like that, you can store these things. But still cyclophilic bacteria can grow on under refrigerator too, remember. But normally in our environment, in environment like India, where the normal or room temperature is almost average temperature is 30 degrees Celsius, there we cannot find more of those cyclophilic bacteria. But in the countries where the temperature normally belongs to 10 to 15 degrees Celsius, then you can find the cyclophilic bacteria. And in those regions, this refrigerated condition needs to be more, a little bit less temperature than this 5 degrees Celsius temperature. Okay. Now let's talk about the microorganism growth in food. So what are the factors that govern the growth of food? We have talked about all these factors. So these factors are called intrinsic factors and extrinsic factors. Extrinsic factors are the factors which come from the outside of the food, not present in the inside of the food. For example, temperature, relative humidity, the contaminated microorganisms, so the content of microorganisms, all these things come from the outside of the food. So these are the extrinsic factors which controls the microorganisms growth. and which is the intrinsic factor. Intrinsic factor is the factor of the food, like that composition of the food, the water content of the food, the pH of the food, the physical and biological state, and the appearance of the food, and all these things is a property of a food. That's why it is called the intrinsic factors. So whenever we change in extrinsic factor or intrinsic factor, then you can change the microbial community or microbial population. So if we change the population of microbes, then you finally make a change in all these factors, and finally it leads to the damage of our foods. Okay. Now let's talk about this intrinsic factor. We have talked about the composition of the food. So what the food is made up with. The food can be made up with different protein contents. It will be made up with carbohydrate, it can be made up with lipids or fats. If it is made up with protein contents, the microorganisms will be there to take this protein, and in those foods, and those foods we call and normally the division and the breaks down of protein can cause different damages in that food. How would be the damages? Because protein is a structural component. It can be a structural component of a food, and when it is a structural component of a food, when a microbial enzyme attacks this protein and the protein damages, then it ends up with different odor, and we call them the putrefaction. We call them putrefaction. So this is the term which is used so really bad handwriting. I'm sorry, very sorry for the PUTRIFACTION. This is a putrefaction. That means the damage of proteins found in food by the microorganisms. Okay. And also the carbohydrates can be damaged. The carbohydrates can be damaged, and we call it fermentation. Normally we are really familiar with that. When the carbon dioxide is damaged by different microorganisms, different anaerobic microorganisms, we call them fermentation. Okay. And if we damage this lipid or the fat portions, we can make this damage, and we will form fatty acid and glycerol, which are the component of fat, and that will make this fat really odorous and all this. So if we damage this fat, that will be really noxious, and really bad smell is coming from the food when the fat is degraded. Okay, so these are the different types of reactions that can go on when the food is breaking down by those bacteria. Okay. So that's why the food composition is really important. If you have carbohydrates, we can form the fermentation. As a result of it, most of the time when carbohydrate containing food are digested by bacteria, it forms various bubbles, or we can say different bubbles or foams like this. Foaming or bubbling will occur here in this case of thing. Okay, when a proteinaceous food is damaged, it produces noxious odor by this putrefaction reaction. Forms a noxious odor and very bad smell and very bad taste to it. And when the fatty acid is breaking down, then it also forms a fumigation, and also it again produces just noxious smell and all these things. Okay, so sometimes the carbohydrate when it's fermented can be eaten, but the protein, when the protein product is damaged, or the lipid product is damaged, it cannot be eaten because they are really bad. Okay, the hydrolysis of fat can be happen, putrefaction of protein can be happen, and the fermentation of carbohydrate can be happen during the food spoilage. Now pH is also important. We have talked about it before. The presence of availability of water, that's an important content. Low water available in the foods, no problem, but higher water availability, it's always a hard problem. Oxidation reduction potential, this is a potential for the microorganisms. So if the microorganisms have a huge amount of oxidation reduction potential, so if there's some enzymes which can do this oxidation reduction really well, then this redox reaction can damage this food really quickly. Okay, and this oxidation reduction potential of food, the food particles will be really altered when it is cooked. Okay, now let's talk about physical structure. We have talked about that, the presence of antimicrobial substances, that's also an important content. So if we have a food and also the food is contaminated with some bacteria, then if we trigger the temperature, if we add water, then this bacterial culture will grow really rapidly. So the content of bacteria is really important among the food, because food is always surrounding by air, and air is full of microorganisms. As we know, air is full in different organisms, microorganisms, and those microorganisms can sit on this food, and if we do not rinse them well, if we do not make those foods stored in a good place, then this food can be damaged. So normally a food particle to be damaged, it needs many amount of bacteria to damage that food, because those bacteria, because the food is a small particle, and to have a visible damage on it, we need a lots of bacteria to work together. If we do not have a many bacteria, then the visible damage is not, the damage is not visible enough. Okay, now let's talk about the extrinsic factors. In the extrinsic factor, we have temperature. If we lower the temperature, which retard the microbial growth. Relative humidity is another important point. So the higher levels of relative humidity means the higher water content in air, that helps the bacteria to grow. And also the atmospheric condition, for example the oxygen promotes growth, as we have told, and if the oxygen content is going low, some of the other bacteria come on, but there are really less amount of anaerobic bacteria than the aerobic bacteria in normal environment, because normal environment is full of airs. So the anaerobic bacteria are only found in really anaerobic places, but we cannot generally find anaerobic bacteria in our this atmospheric content. If we make this food stored in anaerobic conditions, then it's a good job for us to prevent those food to be spoiled. Okay, we can use vacuum technologies to seal the can, or we can use this canning to make pack the food in controlled atmospheres without